Hi, I'm Dr. Ikechukwu Benjamin Moses. Today, I'll be teaching pathogenic bacteriology, that is AMB 323 for microbiology students. Uh, as a follow-up from my introductory video, today the topic that I'll be talking about is more like a question, and the topic is what makes a bacterium pathogenic? Here is my lecture presentation outline. I'll be defining some important terms: pathogen, pathogenicity, virulence, and I'll also be talking about pathogenic arsenals of bacteria, genetic basis for pathogenicity, bacteria pathogenesis and host relationship, major characteristics of pathogenic bacteria, bacteria pathogenesis and antimicrobial resistance, which is actually a growing and trending public health problem today. I also talk about how bacterial pathogens evolve and where they actually come from. So definition of important terms. Uh, pathogen. Pathogen is a microbe or a microorganism that actually has the ability to cause disease or a damage in their host organism. And the host organism could actually be animals, they could be plants, they could be humans. So in this context, when you say a bacterial pathogen is just a bacterium that has the ability to infect and cause disease in their host. So pathogenicity, on the other hand, when you compare it with virulence, they look similar, but there's some little level of difference in the sense that pathogenicity is just the ability of a microbe to cause diseases, whereas virulence is just a form of measurement, a measure of the pathogenicity, that is the relative capacity of a microbe to actually cause damage in a host. The next thing is pathogenic or virulence arsenals of bacteria. Here, I'll be talking about the weapons that these bacteria possess for them to attack pathogenic bacteria. So, in order for a bacterium to actually be pathogenic or virulent, it must have the ability or it must have capability that allow it to infect the host. So, collectively, all these capabilities, all these arsenals or weapons, they call them pathogenic or virulence factors. And and these factors actually help them to invade the host and also helps them to escape the host defense mechanism because the host also fights back with its immune system, and also it also helps them to cause infection or disease, which could actually lead to death of the host eventually, possibly in extreme cases. And some of these pathogenic arsenals, or let me say pathogenic factors, are actually grouped into different categories. We have the adherence factors, we have the invasion or spreading factors, we have the toxins, we also have the siderophores. And here is the adherence factors. When you talk about the adherence factor, from the word adherence, adhere means to attach. So these are factors that help the bacterial cell to attach to the host cell, either attaching to cause infection in the host or maybe helping it to escape the defense mechanism of the host. For example, this is an endocytic pattern recognition receptor. So with the position of the capsule by this bacterial cell, it's able to escape the receptors of this immune cell that is actually supposed to attach to the receptor here. And we also have the slime layer, we have the pillar, we have the flagella. There's a lot of organisms that actually possess some of these features that help them to invade the host. And this is also a capsule. Here, the dark region, the dark area here is the capsule. So this picture was taken with an electron microscope. That's why you see this dark background like this. And examples of bacteria with these weapons, the capsule, the slime layer, the pillar and flagella, we have the Vibrio cholerae, we have the E. coli, we have the Salmonella, we have the Neisseria gonorrhoeae, we also have the Streptococcus pyogenes. Now, next is the invasion or spreading factors. So, invasion actually is just the ability of a pathogen to spread to other locations in the host by invading the host cell, while the spreading factors are just they are chemicals released by bacterial cells, and these chemicals are called enzymes. So, what these enzymes do is that they affect or they attack the physical integrity of the host cell of the host cell tissue matrices and even intercellular species. So, this actually helps the bacterial pathogen to spread all throughout the host. And examples of these bacterial enzymes, we have the collagenase produced by Clostridium species, we have the neuraminidase, we have the streptokinase, we also have the staphylokinase, which is produced by Staphylococcus species, and we also have the leucocidase and the hemolysates. And the next interesting thing I would like to talk about is the toxins. Toxins are more like poisons for the host, which are actually produced by the bacteria. So toxins are more like a very specific virulence factor actually produced by some bacterial pathogens. And most of these toxins actually have been known to cause diseases in humans. We have some of them that cause botulism. Botulism is actually caused by Clostridium botulinum toxin. We also have the tetanus, we have the diphtheria, we have the diarrhea. And there are actually two major categories of toxins. We have the exotoxins, we also have the endotoxins. From the word exo and endo, exo means to be released to the outside environment or surrounding environment, while endo retains or stays inside of the particular pathogen. So exotoxins, that's why I said exotoxins are protein toxins that are released from viable bacteria into the surrounding environment, whereas endotoxins are toxic lipopolysaccharides made by gram-negative bacteria, and they are more sometimes even a component of the outer membrane. So they can only be released, for example, the endotoxins can only be released when there's a lysis of the bacterial cell, or maybe when the bacteria actually dies. Then the endotoxins embedded in this outer membrane then gets leaked out to the environment and can actually cause disease in humans. The next one is I'm going to talk about the siderophos. Siderophos is very interesting because it centers around iron. Iron is an essential growth factor for host cells. So what happens is that some of these bacterial pathogens, for example, the E. coli, the Salmonella, we even have the Neisseria, what they do is that they have some specific receptors that attach to the host cell. And when they attach to the host cell, this particular receptor gives a kind of signal to compete for the limiting nutrient, which is iron, because this iron is required by the host to actually produce hemoglobin or produce or assemble hemoglobin, transferrin, and lactoferrin. So, and when this iron is being competed with by these bacteria, invading bacterial pathogens, it actually starves the host cells for all this iron, which is an essential component to produce all this hemoglobin and the rest. So when that happens, whatever that is produced is not going to be efficient enough, and that can actually lead to the death of the host cell. So an example of a bacterial siderophore is enterocolin, which is produced by E. coli and Salmonella species. This genetic basis of for pathogenicity or virulence, virulence or virulence. Actually, in this context, we talk about pathogenic factors in bacteria may be encoded on the chromosome. It could be by bacteriophage DNA. It could be on the plasmid or transposon. And novel pathogens are actually evolving, or let me say, constantly emerge as a result of competitive selective pressure, which they find themselves in different environments. And in the process of trying to adjust, adapt, they are actually evolving to new strains or emerging strains. And most virulent strains did not actually originally intend, they don't actually have intention to damage the host cell, but rather it's more like a kind of competition with other microbes in order to survive or obtain nutrients from the host. And if you look at this diagram here, you see this is an avirulent E. coli. Suddenly becomes a virulent E. coli. So what happens is that here it is not pathogenic, but here it's pathogenic because it's able to possibly it actually picks up some genetic material through conjugation, transduction, the transformation, and change and transformed to a virulent strain. Next thing I will talk about is bacterial pathogenesis and host relationship. So pathogenicity is just a multifactorial process which actually depends on some important factors. It depends on the number of organisms in the exposure, that is the bacterial inoculum load, also the nature of the bacterial species, and also the immune system of the host. Is the host immunocompromised? Is the host immunosuppressed, or is the host immunocompetent? So these are factors that play out in the manifestation of a disease due to pathogenic bacteria. So to be able to effectively invade or infect an organism, the pathogenic microbe or microorganism must be able to overcome all the host defense mechanisms and other attributes. The major characteristics of pathogenic bacteria are infectivity, virulence. I've already mentioned some of these things in my previous slides. Transmissibility, adaptability, must be able to adapt, reproduction, specificity, and evasion of host defense mechanism, toxigenicity, incubation period, and the genetic variation. And the next thing I'm talking about is bacterial pathogenesis and antibiotic resistance, which is a growing and trending public health problem. So, well, antibiotic resistance has actually become a serious global public health problem, which an estimated death of 10 million annually worldwide by the year 2050. That is a report by Jim O'Neill in 2016. He made a report for the WHO. And over the years, actually, there has been progress in the production of antibiotics, for example. But the problem is that uh, due to antibiotic misuse and abuse, there are several worrying setbacks. So now a lot of infections or a lot of bacterial drug resistant strains are emerging and re-emerging, and even all diseases that were known to actually have been eradicated, like tuberculosis, anthrax, and the rest, they are now suddenly coming back. Like they have been, like the report last year in 2023 reported that in 5 countries in the East and Southern Africa region recorded anthrax outbreak, with more than 1,100 suspected cases and 20 related deaths. So this is actually very, very worrisome. And the next thing I'm talking about is how do bacterial pathogens evolve and where do they actually come from? So they evolve through selective pressure, because when they find themselves in different environments, either good or bad, they try to survive, they try to adapt. So in this process of adjusting, they evolve, and the pressure of the environment helps them to evolve into a new strain that actually survive very harsh conditions, just like the same way we human beings survive or adjust to the cold weather, to the hot weather. And the next thing is mutations. Mutation is just a change in genetic material, or let me call it a form of mistake. This actually happens during reproduction, or just like we see in humans. Like for example, in human beings, we see sometimes we have this birth defect. So just like a kind of mistake in the genetic sequence of the bacterial cell when they are trying to reproduce, or sometimes it can even be environmentally induced. Maybe there is kind of a ray of light actually, or maybe a particular environment actually induce this and actually become genetic and get transferred to another strain or the offspring. And the next one is gene transfer, which I already talked about: conjugation, transduction, and transformation. And conjugation is just an exchange of genetic material between two different bacterial species. While transduction is just, in this case, a virus picks up this genetic material in the environment. The genetic material could be antimicrobial resistant gene, and then transfers it to the bacterial pathogen. And in the case of transformation, the bacteria just wallows around in the environment and just picks up genetic material, which will actually offer a survival advantage to the bacterial cell. So this is the end of my lecture. Thank you very much, and I look forward to seeing you in my next class. Thank you.